days everyone. For this video, we will discuss details of the mechanisms to transport our copper ion from the food that we consume to the benefits to our human body. Okay, so as it's written in the report, the copper ion that we consume from food, it can be from organ, meat, shellfish, and nuts. For examples, cashew nuts, hazelnuts, almonds, salmon fish, crab, prawns, and animal livers. We can consume the copper ion from this food because this food is rich in the copper ion micronutrients. Okay, when it's consumed from the food, it will get absorbed by our body. Where it will get absorbed in the small intestines. The large intestines is to absorb water, the excess water. For small intestines is to absorb the micronutrients and macronutrients. Okay. When after the copper ions is absorbed by our small intestines, it will get transported via the portal veins to the sinusoid. To the sinusoid near where? Near the hepatocyte. Near the hepatocyte. Okay, as the known uh, as here we already label hepatocyte also known as liver cells, which means the whole process is occurring undergo under in the liver. Okay, so the copper ions will stay in the sinusoid. Then it should be go where? It should go to the Golgi apparatus. Okay, before it go to the Golgi apparatus, it should go to the matrix first, which means outside of the sinusoid, between the sinusoid and the Golgi apparatus. So how they get transported out from the sinusoid? By one of the protein, we call it CTR1, which is the copper transporter 1. This copper transporter 1 here. Okay, why I will give the name of the copper transporter 1? Because in the sinusoid they have a lot of other transporters, not only one copper transporter, so we label as CTR1. Okay, the CTR1 will transport the copper ions to the matrix. After it's transport to the matrix, then it should go into the Golgi apparatus. But here we have a barrier. We call it ATP7B protein. This is one of the uh, we can say the door to enter the Golgi apparatus. In here, I just only draw one, but actually in the on the membrane of the Golgi apparatus, there's a lot, but I just draw one for easy explanation. Okay, this Golgi apparatus, okay, this ATP7B proteins is used for what? It's used to regulate the copper hemostasis in the Golgi apparatus, which means what? It will only pump in the copper ions that needed into the Golgi apparatus, and others that not needed, or in other words, excess copper ions, the Golgi apparatus will pump out to the bile duct. Okay, now we will talk about the excess copper ion first. When it's pumped out to the bile duct, when we talk about bile duct, we will, in our mind, it will link with urinary bladder. Why? Because bile ducts will excrete, not excrete lah, transport the excess copper ions to the urinary bladder and to produce the urine, to use the urine to excrete all of the excess copper ion. Actually, not only excess copper ion, it's only included magnesium and sodium. This is why your urine have a slightly salty taste. Okay. After we talk about the excess copper ion, how about we talk about the copper ions that already go in to the Golgi apparatus? When the copper ions go into the Golgi apparatus, it will bind it to two things. First is the apoceruloplasmin, and the second is albumin. But for this video, I will focus on apoceruloplasmin and ceruloplasmin because I will use these two carrier protein as my examples for the further mechanisms, for the further metabolic activities, in other words. Okay, so before we going through further the mechanisms, we need to know what is apoceruloplasmin first. Okay, apoceruloplasmin, if we take a look on the nomenclature, is like a hexagon. When it is like a hexagon, it can bind with 6 of the copper's ion. After they bind with the copper ions, they will have a new name. We call it ceruloplasmin. Okay. When it's already become ceruloplasmin, the carrier protein, this ceruloplasmin, it will carry these copper ions to other organs. For example, the bone marrows for hemoglobin synthesis, the brain cells for the formations of the nerve cells and connective tissues to form the cross-linker collagen. Okay. Now I will use two of the examples. Before I use the further examples, we will talk about the redox reaction. Okay. This is the redox reaction of the copper ion, and it's very important why it will use as a catalyst in the further mechanisms in other metabolic activities. So for our first examples, we will take a look at this diagram. I will call it ETC. In long term, we call it respiratory electron transport change. Transport chain. Okay. Actually, it's occur in the mitochondria. Okay. In here, we know that we saw that we have complex 1, complex 2, complex 3, complex 4, and complex 5. Okay. Complex 5 we also known as ATP synthase. Okay. Where is the copper used? Copper ion used. Okay. Here I already label it out. It will use in the complex 4, the cytochrome C oxidase. But from this diagram, we can't know where is the copper. You already say it will use the copper ion, but I didn't see any copper ion label. Okay. If we enlarge the complex 4, you will see something like this. Okay. We we know that here we have two copper ion, the CuA and CuB here. I enlarge here CuA and CuB. Okay. We also can see there is electron, free electron. So the purpose of the copper ion in this cytochrome C oxidase is for electron transfer for ATP synthase. Okay. Now we go to second examples, hemoglobin synthesis. Okay. Maybe others with general knowledge will ask, hemoglobin synthesis, I think, is just only involved with iron. What's the matter with the copper ions? Okay, don't worry, we will go one by one. Okay, when we consume the iron micronutrients from the food, it will also going through the same absorptions by the small intestines to others. Okay, now before it's used for hemoglobin synthesis, it should be bind to the transport protein because it transferring first. It's very important because it's the carrier protein to form the hemoglobin. No form synthesis the hemoglobin. Okay, so what's the functions of the copper ion in here? Okay, the ion that we consume and absorb by our body, it is not really can bind with the transport protein transferring. So we need ferrosidase. In other words, it's also called as ceruloplasmin. It will oxidize the ion from divalent states to trivalent states, so it can bind with the transferrin to synthesis the hemoglobin. Okay. From here, we know that the ferrosidase, that the prefix is ferro, which means the iron, then oxidase. So when we combine ferrosidase, it is to oxidase the iron. Okay. Usually, when we touch about the general knowledge of hemoglobin synthesis, we just only know the ferrosidase. We don't know the ceruloplasmin. If we know that ferrosidase is equals to ceruloplasmin, then it will be easier to knowing it. Okay. Then we have a question for here. Is it the excess copper will lead toxic to our body? For this question, the answer is yes. But is it will have a high chance to have the copper toxication? No, is a real cases. Okay, why? Because our body actually they can handle to excrete all of the excess copper from our body by urination, by urine. Okay, in what situations we can't excrete all of the copper? When either you consume a lot of copper, something like you eat a whole ball of the raw material copper. Nah, just kidding. Okay. The cases is that the ATP7B protein is dysfunctional, which means it can't pump in the copper ions from the matrix into the Golgi apparatus. So there are no, cannot say no, the amount of the ceruloplasmin form will be very, very less. Then it will have what bad effect? It will have the bad effect of causing amnesia. Why it will causing amnesia? Because if you are not enough ceruloplasmin, you can't oxidize the iron ion. So the hemoglobin that synthesis will decrease. In other words, blood production decrease. And it also will affect your nerve cells and your connective tissues. Okay. Then after we talk about dysfunction of the ATP7B protein, okay, it will lead to this. But the factor to lead this situation also have another, which is less consume of copper. If there is excess of copper ion that consume and your ATP7B protein is dysfunctional, then all of the copper ions will transported to the bile duct. When it go to a bile duct too much over the working amount, then the bile duct will start to dysfunctional. When this dysfunctional, it can't let the urinary bladder to excrete out all of the excess copper ion. At the end, it will it will lead to the copper toxication and it will damage the liver. Okay. After we talk about this, we will talk about the daily intakes guideline for the copper ions. We can go to the site of the National Institute of Health of the United States. In this website, they have the table of contents. Okay, we just go to the recommended intakes. In this table, it shows that above 19 years,